For you, sorry, it's good evening to you. Good evening, everyone. It's early morning here.
Good evening, sir.
Thank you. I sincerely thank this opportunity. And today we'll be discussing novel strategies to prevent and treat neurodegenerative diseases. So that's the topic that we are going to talk about. So to start with, let's again first understand why we are focusing on neurodegeneration. There are two major concepts when you think about anything in the brain. There are two types of cells in the brain. The brain cells can be classified as neurons and glial cells. So those are the two types of cells in the brain. So whenever we think about neurodegenerative diseases, that there is no problem associated with the glial cells. It's always the neurons. So when you think about, again, what is happening to the neurons in neurodegenerative diseases, two things happen. Number one, you have an irreversible neuronal loss. Most of the other body tissues or cells, they can be replaced. For example, if you have a skin burn, if you have an injury to the skin, am I right? What's going to happen? Those can be replaced. However, the neurons, whenever we think about neurodegenerative diseases, the neuronal loss is irreversible. That's the first concept. The second concept is it is progressive. What do you mean by progressive? So, which means when the patient is around 60 years old, the patient may have 1,000 neurons. I'm just giving you an example. But during the progression of the disease, you can see from this particular slide, what happens is there is an increased loss of neurons. There is an increased neuronal loss. So whenever you think about any neurodegenerative diseases, the two major concepts are irreversible, progressive. Okay, that's very good. So now what can we do? We cannot treat these diseases. So the big picture here that we are going to take home is neurodegenerative diseases cannot be treated. But we can have some strategies to prevent or slow down. The better word will be to prevent or slow down the loss of neurons. Okay, so the concept. The third concept, what are the most prevalent neurodegenerative diseases? At least we are talking to the pharmacy students. Most of the curriculum, we teach Parkinson disease, Alzheimer's disease, and we teach Huntington disease. Very good. So now, when we think about these, we think about these are the most prevalent. Now, what is the third concept we have to take home? The irreversible and progressive neuronal loss in each of the disease occurs at a very specific part of the brain. So in Parkinson disease, it is substantia nigra. In Alzheimer's disease, it is basalis minor, hippocampus, and cortex. So now, the big picture that we are taking home from this particular aspect is progressive, irreversible neuronal loss occurring at a specific region of the brain or occurring in a specific region of the brain. So that is the most important thing we want to understand. Good. So now what do we want to do? We want to prevent or decrease or slow down or have a break in the loss of the neuron. How can we do that? So there are several models. So you can have an in silico model. That's what we are going to talk about today. How valuable in silico model are. In vitro model. Then we have ex situ, in situ, ex vivo, in vivo. So ex situ, in situ, and ex vivo, much of them are not very popular. They are catching up, but very popular currently are the in silico, in vitro, and in vivo, which I do it in our lab. So I'm going to talk about those kind of techniques. So these are the models that are currently associated with drug design to prevent neurodegeneration. Very good. So now in our lab or in this particular study, we are going to focus on three major topics. We are going to talk about in silico studies, in vitro, in vivo. When we used to be in pharmacy school, I'm talking about 80s and 90s, I was when Dr. Jabal and G, when we used to have chemistry of synthetic drugs, chemistry of natural products. So all those subjects combined are now this particular topics in silico study. So in an in silico study, I'm going to talk about that. Based on the structure of the compound, we can determine both the pharmacokinetic and the pharmacodynamic property of the drug. So try to understand that. When we think about first pharmacokinetic, what do we mean that? Absorption, distribution, metabolism, and elimination, we can determine. So we can say whether the drug is going to act centrally or peripherally. What is the duration of the drug action? How is the drug going to be metabolized? How is the drug going to be eliminated? So from a pharmacist point of view, this is very important. For example, what are the possible drug interactions? We may not be playing a very important role with regard to the pharmacology or prescription of the drugs. But currently, with the new PharmD curriculum where we are going is, we are the ones who are going to monitor how the drug is acting in the body. We are going to look into the pharmacogenomic effect. It's a big field. It's a very big field. Now we can carry that. Currently in India, because of COVID, we are having a lot of apparatus, a lot of toys, a lot of scientific tools that people are using to identify COVID. All those equipments, the RT-PCRs that we have, can be used, right, to see whether a patient is vulnerable to a neurodegenerative disease. We can do a lot of genomic research with all the equipment we have. So that's the most important thing. I'm going to ask Dr. Jayavalanji and the dean to make sure that we have those kind of instruments where we can do genetic testing in a lot of patients and we can have data and predict what is happening in Rajasthan with regard to neurodegenerative disorder. So it's a very big thing that you guys are sitting on a gold mine now. You just have to think about future collaboration and go from there. But then, so in an in silico study itself, you can identify a compound, whether the compound is good enough to use for the treatment of neurodegenerative disorder. And then we can do an in vitro study. We can use a proper cell line in vitro, not in an animal model, outside the body, that is in vitro. We can do it in vitro study. And then we can go to an animal study. After animal study, we can carry on to a human clinical study. So that's the big picture. Everybody knows that. Okay, good enough. So now I'm going to use a very important software, and I'm going to tell you how we can determine the pharmacokinetic aspect of the drug. What do you mean by pharmacokinetic aspect? So initially, we can see what are the can the drug be absorbed in the body? Can the drug be absorbed orally in the body? Okay, how is the drug going to be distributed? Is it going to be distributed only in the peripheral system, or can the drug cross the blood-brain barrier? Does the drug have enough lipophilicity to cross the blood-brain barrier? And we know that it's not always the lipophilicity. Why am I talking about that? Think about the blood glucose. Glucose has to go to the brain. It's not a lipophilic molecule, which means it has a carrier or it has a transporter that allows the molecule to be crossing the blood-brain barrier and entering the central nervous system. So think about those kind of aspects. How can we design? So for any drug discovery, okay, we have to understand that we now should have an in silico model. All these softwares are available. There are plenty of softwares where we can do the pharmacokinetic and pharmacodynamic property to validate our in vitro and in vivo profile. Very good. We are on the same page. But these are very expensive. But from a school point of view, if you are associated with a drug design, try to understand and get this. This is a very good thing that we have to teach our students. Maybe when you design a new curriculum, Jayavalanji or the dean, I just want you to understand a novel drug design course. Try to have that, please. That's a very good aspect for all the young students to carry on into their pharmacy professional career. So that's one good aspect that we can think about. So we use QuickPro. So I'm going to go and show you some data. This is an old wine in a new bottle. This has been the one that we have been using for 60, 70 years. The area under the curve, the relative bioavailability, Cmax, Tmax, T-half. So these are the pharmacokinetic parameters that we have been using for the last 40, 50 years. But this is not sufficient. So when you're going for FDA approval, we are going for that. Yes, this is very important. I'm not telling away that. This is very important for us to have these data. But in a bigger picture, is this data sufficient enough? So now, we recently published a paper in Frontiers in Pharmacology. This is with a collaboration from an Indian scientist, one of my good friend, whom I have known her for 30, 35 years. We have been collaborating on this project. So the most important thing, this particular parameters that we have, the traditional pharmacokinetic parameters, we need them. I'm not denying the fact. But now, when you think in a bigger picture, can we make some hybrid molecules? So this project was about Piper longum, our pepper. So how can we increase the bioavailability of the pepper? Okay, or how can I increase the bioavailability of boswellic acid? Again, a very traditional Indian origin substance. Boswellic acid has shown to have anti-inflammatory effect. So now when you think about, I'm just going to throw you an example. Now you think about COVID, am I right? So now please somebody let me know, what is the pathology of COVID? We all accept that COVID occurs because COVID is an infectious disease. We don't have a problem with that at all. But the problem is, it is not the infection that is causing all the symptoms and all the morbidity and mortality. The big word, morbidity and mortality, the disease and the death. It's not just the infection. What is happening? It is the inflammation. They call that as cytokine storm. Whenever you think the word COVID-19, the problem is cytokine storm. What do you mean by cytokines? There are a lot of chemicals because of COVID infection. A lot of chemicals are being released in the body. When a chemical is being released from the cell, that is known as cytokine. Cyto means cells, kines means chemical. So these are pro-inflammatory cytokines. These can damage all the organs in the body. The most important organ associated with COVID-related death is the respiratory tract, the lungs. Am I right? So you think about that. So now if we can have a very potent anti-inflammatory compound with us, it's going to decrease the COVID. So in a bigger picture, let's think about that. Relatively, the number of deaths, the morbidity and mortality associated with COVID-19 in India is very low. Now let's ask the question, why is that? Maybe in the everyday food that we eat or we consume, like pepper, your turmeric, your curcumin, all those have significant anti-inflammatory effect. So now if we go with the hypothesis that COVID-19 is associated with infection and inflammation, the diet that we are consuming or in India is most of them have anti-inflammatory effect. So that may be a reason that we have less morbidity and mortality associated with COVID-19. So again, I'm coming with the novel strategies, how we can reduce this kind of neurodegeneration. In general, in India, we have less neurodegeneration as compared to the Western countries. Maybe again, it's not only the diet, think about that, genetics too. So we designed some compound based on the piper and boswellic acid. And then we did a quick pro in silico study. So the concept that we found was, yes, this drug can be administered. These drugs that we design as hybrids, they can be administered orally. They can cross the CN, they can cross the blood-brain barrier. You can see that. Okay, that's again pertaining to pharmacology. Now let's come to a bigger picture. As a pharmacist, what is more important? How is the drug being absorbed, distributed, metabolized, and eliminated? Think about the metabolites. So for everybody in the pharmacy, whenever we take a drug, a medicine, the drug has to be metabolized. Do we have an active metabolite? So again, a very important thing. Whenever we study about a drug, we not only study about the drug, how the drug is being carried out in the body. Do we have an active metabolite? How many active metabolites do we have? How many metabolites do we have? What happens to the metabolite? Are they staying in the body? Are they having a hypersensitive reaction? Are they readily being eliminated from the body? So all those kind of questions comes. So with this model, we not only can predict the effect on the CNS, oral administration, but we also can see how many metabolites, possible metabolites that is generated by this particular drug. Okay. Then with the same thing as I told you, whether how much do we have oral absorption? So you can see that the hybrids that we designed, why did we design these kind of hybrids? So think about that. The boswellic acid derivatives, they have only 80 to 73. The lowest was 73, and then the highest was around 96.2. So when we modify the structure, we have an idea by modification of hybrid molecule, we can have a drug that can be 100% orally absorbed. That's number one. Number two, we also compared with aspirin because aspirin is a well-known anti-inflammatory drug. So we just want to throw in aspirin and see what is happening. So always we have to make sure that we compare with the existing accepted well-known drug. So we did with aspirin. So again, this quick pro model is a great model to understand. It's a kind of a strategy for us to design novel drug design. So again, we had another parameter that we looked into that to validate the drug not only can be administered orally, but it also can have effect in the cellular system. And then we didn't leave it there. So now what did we think? Okay, let's see. We will administer boswellic acid. And then we had a formulation of boswellic acid with piperlongum extract. And you can see that when we administer to the rabbit, the amount of boswellic acid in the serum was increased, by around 20 to 25%. And the effect was also prolonged. So from a pharmacokinetic and pharmacodynamic point of view, try to understand that we are going to have the effect of the drug for a longer duration at a higher bioavailability. So that helps us to understand how in silico study can validate our in vivo finding. So that was the first study. And I just want to let you know that we had published this particular article in Frontiers in Pharmacology. So I just want to let you know that how we ended up. So now that's the first strategy. Okay, now let's think about the next one. Okay, so I'm going to go So whenever we think about, again, what are the strategy to reduce neurodegeneration, we want to identify a pattern. Okay, so what we did is we identify serotonin. So serotonin, okay, it's a big family. There are 7 members of serotonin. There are serotonin 1, serotonin 2, serotonin 3, serotonin 4, 5, 6. It's a protein signaling molecule. If you're try to understand, most of the signaling molecules can be classified based on their structure. Lipid-based signaling molecule, all the icosanoids that we are getting, right? All the isoconoids that we are getting, that's the prostaglandins, the lipoxin, leukotrienes, all those things, they are derived from they are lipid mediators. But serotonins are proteins. They are proteins that affects the metabolic regulation. So there are 7 serotonins. We are mainly focused on SERT3. Why is that? Because it is associated with beta oxidation, fatty acid oxidation, amino acid oxidation. It helps or increases the mitochondrial function. Whenever any protein increases the mitochondrial function, what is the net effect? It increases the production of ATP. Okay, that is the most important. So why are we talking about ATP? Whatever we consume, whatever we eat is ultimately converted to ATP. ATP is the sole energy for the body, right? So we are understanding. So we are thinking about SERT3 is directly associated with the mitochondrial function. And not only that, SERT3 has antioxidant effect. What do you mean by that? It can scavenge reactive oxygen species. Thereby, it can decrease programmed cell death. What do you mean by programmed cell death? That is apoptosis. So in a bigger picture, a drug or a molecule that can enhance the effect of SERT3 can increase the mitochondrial function, decrease oxidative stress, decrease apoptosis, and increase the neuronal survival. So now when we think about neurodegeneration, what is the big aspect that we are trying to say? Anything that can increase the neuronal survival. So SERT3 can be one mechanism associated with that. So again, in a bigger picture, SERT3 is not only associated with energetic status, okay, regulation of the energy due to mitochondrial biogenesis or increased mitochondrial production. However, it is associated with cancer. Okay, the main picture is it is associated with cancer. You can see that. And then it is associated with cell signaling, numerous cell signaling. You can see that. So it's associated with cancer, cardiac function in the brain. So a lot of research has been done on SERT3. Okay, very good. So we took SERT3. Okay, one more strategy. But initially, again, now we are going to another software where we can study about the docking images. Okay, it is also a quick pro. But now try to understand if SERT3 is a protein, the next thing is, can a drug bind or interact with the protein? So Honeckall, again, it's a botanical based compound. Okay, it's a natural product in a bigger picture. Honeckall, what does it do? Honeckall, you can see that CNS, you have a number 0, -2 to +2. Any drug with the value of that can have an effect in the cell nervous system. It can cross the blood-brain barrier, it can have that. So we have a docking score. A score with the minus, right? It shows that it has a good binding affinity. It has a good binding affinity. Very good. So now definitely we can understand that Honeckall, you can see that a natural product can bind or can associate itself with SERT3. So that's happening. So we can see that. Very good. So you can see that it has a binding. So what we did was, we did for 7 compounds. Okay, we had trilobatin. So we had epsilon, dihydromercetine, adjudin, and salitroside. So all these compounds, we did it, and we found that they have an affinity . Can you hear me?
Yes, sir.
Okay, I'm sorry. Yeah, somehow we got lost. Okay, good. So now we are all on the same page. Okay, so now we made sure that we have a drug, and we made sure that this particular drug has a binding affinity to the SERT3. Why are we studying about SERT3? We know that if we can increase the function of SERT3, what happens? We are going to have increased mitochondrial function, more ATP, decreased reactive oxygen species, decreased oxidative stress, decreased programmed cell death. Therefore, the neurons are going to survive more. So that is what we want to do. With regard to neurodegeneration, what do we want to do? We want to increase the survival of the neuron or decrease the neuronal death. So we thought that SERT3 will be a good one, and then we identified Honeckall can be a good drug. So based on our finding, what are we going to do? So now, based on that, so we recently published that. So nutraceuticals-based SERT activators as a therapeutic targets for Alzheimer's disease. Okay, big picture. Very good. Whenever we think about Alzheimer's disease, what is the most important thing that comes to our mind? Number one, cognitive impairment. Very good. Number two, what is happening? We have a particular protein. What is that protein? Beta amyloid. So you have increased deposition of beta amyloid that causes neuronal death. So based on the concept, what are the causes of neuronal death? Okay, beta amyloid is there. Now, how does that relate to the neuronal death in Alzheimer's disease? The neuronal death in Alzheimer's disease usually is associated with apoptosis. You have inflammation. We have oxidative stress, excitotoxicity, and mitochondrial dysfunction. So if you think about a bigger picture, okay, we have beta amyloid deposition, very good. And top of it, what's happening? We are having oxidative stress, inflammation, apoptosis, excitotoxicity, and mitochondrial dysfunction. So now, for people who are students, we have try to understand what is oxidative stress. So you have more pro-oxidants and less antioxidants. So there's a balance between pro-oxidants and antioxidants. When you have increased pro-oxidants and decreased antioxidants, you have oxidative stress. There are various markers associated with oxidative stress. You have increased reactive oxygen generation, increased lipid pro-oxidation, depletion of the pro-oxidant glutathione. I'm just giving you a few examples. Okay. Whenever you think about apoptosis means programmed cell death. So you have breakdown of the DNA. How does the breakdown of DNA occur? Maybe because of caspase activation. Caspase 3 is a protein that can cleave the DNA and cause DNA degradation. So that is apoptosis. Mitochondrial dysfunction. So with regard to mitochondria, you know that there's an electron. It's an electron transport chain is there, where you have complex 1, complex 2, complex 3, complex 4, where the electrons are being transferred. And during that process, there's a redox reaction occurring. And then ADP is converted to ATP. So there can be a decrease in the complex 1, 2, 3, 4 activities. That can lead to depletion of ATP. That is mitochondrial dysfunction. Inflammation is very similar to your oxidative stress. There's a balance between pro-inflammatory mediators and anti-inflammatory mediators. During inflammation, you have more pro-inflammatory mediators and less anti-inflammatory mediators. As I brought you, currently we are going through a pandemic. And whenever you think about COVID-19, this is what is happening. The pathogen, right? The virus, the COVID, what is it doing? It's causing an increase in the generation of the pro-inflammatory mediators. That's the reason it's called as the increased cytokine storm, okay, associated with COVID. Then the most important thing when you think about Alzheimer's, at least excitotoxicity. Glutamate is the neurotransmitter that binds to the NMDA receptor and increases the calcium influx, leading to neurodegeneration. So we use a NMDA antagonist. So these are the most important mechanisms associated with neurodegeneration. Okay, so you can see that in oxidative stress, we told there's a balance between antioxidant free radicals, and where you have less antioxidants and more free radicals in oxidative stress. Same way, apoptosis, programmed cell death, you can see that caspase activation, degradation of the DNA. That's the whole idea about apoptosis. Then we talked about mitochondria. ATP is the most important thing that is being formed. So during that, what is going to happen? The electron transport chain can be inhibited, leading to depletion of ATP. We talked about mitochondrial dysfunction. The inflammation, very similar to oxidative stress, pro-inflammatory, anti-inflammatory mediators, imbalance. Then we have excitotoxicity. You can see that glutamate acting on NMDA receptor causes calcium influx, leading to excitotoxicity, where the neurons are undergoing neurodegeneration, neuronal death. Okay, so now what did we do? We picked a drug that can enhance or bind to SERT3. I showed you that Pharmacological can bind to SERT3 protein. So what did we do? We took a cell line that produces beta amyloid. So now, why did I bring the point? In Alzheimer's disease, the most important cause of neurodegeneration may be attributed to drugs that enhances beta amyloid production. So now we have what we have. We have a cell, right? PS70 cells, which can produce beta amyloid. So now try to understand that we incubated Pharmacological and we found a dose that does not kill the PS70 cells. So 5 micromolar and 10 micromolar. So what is our goal? Our goal is to make sure that it does not kill the cells, but it only can reduce the beta amyloid. So we took 2 doses, and then you can see that these drugs, again, what was the concept? SERT3 drugs that affects the SERT3 protein or increase SERT3 protein what can it do? It can cause decreased oxidative stress. You can see that Pharmacological, a SIRT3 activator can increase the activity of the antioxidant enzymes, SOD activity, catalase activity, and glutathione peroxidase activity and glutathione reductase activity, which means it's going to reduce the generation of reactive oxygen species. You can see that specifically. We can see that our drug, Pharmacological, a SIRT3 activator, can decrease the generation of lipid, reactive oxygen species. Therefore, it can inhibit the lipid peroxidation. So we can understand that concept. And then you can see that next we want to see what is happening to the mitochondria, because we have been telling that a SIRT3 protein can affect the function of mitochondria, increase the mitochondrial function and increase the production of ATP. Boom, we got that. You can see from that it increases the biogenesis of mitochondria. More mitochondria are being formed. Number one. Number two, because of that, you can see that there's increased complex 1 and complex 4 activity, which means more ATP is going to be generated. So now let's come to beta amyloid. So now we have a concept that SIRT3 can decrease beta amyloid. So yes, SIRT3 can decrease the generation of beta amyloid. You can see that, and we are comparing with insulin, a drug. Okay, why insulin? I'll come to that concept, because glucose levels are increased in most of the patients. Hyperglycemia can be associated with increased beta amyloid production. So we are thinking insulin can be another drug to be compared with. So in a big picture, PS70 cells not only decrease beta amyloid, we were thinking what was the reason behind it? Beta secretase is an enzyme that is associated with the generation of beta amyloid or deposition of beta amyloid. So our drug, Pharmacological, a natural product, is a SIRT3 activator, can inhibit beta secretase and therefore decrease the production or formation of beta amyloid. So that was the methodology. So in a big picture, you can see that Pharmacological can increase, okay, the function, antioxidant function by decreasing the generation of pro-oxidants or increasing the antioxidant effect in the body. And then not only that, what it can do, it can increase the mitochondrial function. Okay, therefore, what it can do, it can increase mitochondrial biogenesis and decrease beta secretase activity, which can lead to decreased beta amyloid production. So that's the big picture. So again, it's one of the neurodegeneration strategy to decrease neurodegeneration. Another one of the strategy to decrease neurodegeneration. We got this particular Pharmacological associate SIRT3 activation published in PLOS ONE. So again, I just want to understand that I'm just telling the young students, whatever you find, be honest with your finding, be rational with your finding, try to understand your hypothesis, what was your aim, and then try to publish in a valid journal. It doesn't matter, try to publish it in a valid journal. Make sure that you document everything when you do an experiment. So now the next aspect quickly I want to go is, so now we talked about some in silico properties. We talked about in vitro. So that's the three things I told you: in vitro, in silico, and now we are coming to in vivo. So now I don't know how most of you, I'm just going to recommend, I'm just going to start with Jay Balaji because I know him for a long time. Make sure that you all do a blood test every 6 months. Once in 6 months, try to do a blood test. Okay, I'm just coming to an in vivo aspect. From in silico, in vitro, I'm trying to get to an in vivo aspect. Whenever we do a blood test, we have total proteins, we do albumin, globulin. All these, am I right, are markers of inflammation, infection, okay, bone marrow problems. Okay, so in a big picture, try to understand that these are telling you something about infection and inflammation in your body. Okay, I've got this published. I'll just let you know that how I'm going. So I'm just going to go through the total protein, albumin, globulin, and then think about blood urea nitrogen. Whenever you think about the blood urea nitrogen, think about, do we have a problem in the liver? Okay, how is it being cleared? How is the urea being produced in the body? So try to understand those kind of big pictures whenever you think about that. Okay, creatinine kinase. What is the level of creatinine kinase? What is the level of LDH, lactate dehydrogenase? What is the level of alanine aminotransferase, aspartate aminotransferase, alkaline phosphatase, okay, bilirubin, creatinine? All those compounds, try to understand that. I'm going to tell you that if you have a condition, okay, where you have hyperglycemia, all these are going to be affected. During hyperglycemia, what is going to happen? All these are going to be affected. I'm going to tell you a story on this particular thing. We generated an animal model, okay, with both STZ, okay, that is what is STZ? It's a toxin, streptozotocin. What does it do? It causes hyperglycemia. It decreases the insulin secretion and causes hyperglycemia. Very good. And then we also, because we are in a pharmacy background, I just picked a drug Okay, we picked a chemotherapeutic drug. Try to understand that. So we had two combinations, chemotherapeutic drug, and we had a drug that induces streptozotocin in an animal model. And then we isolated their blood, or we collected the blood and analyzed that. So now what are we trying to say is, so when there is hyperglycemia, you have increased bilirubin, creatinine, and okay, we all expect hyperglycemia. There's increase in triglycerides level. So now why am I talking about this? So you know that triglycerides increase the risk for stroke. Triglycerides increase the risk for myocardial infarction. Triglycerides can increase the risk for neurological disorder. Same way, hyperglycemia, whenever you think about hyperglycemia, what does it do? It increases the risk for neurodegeneration. So why are we talking about that? So hyperglycemia, it causes impaired neuronal signaling, vascular damage, neuroinflammation. It can lead to tau hyperphosphorylation or beta amyloid formation. So now in a big picture, a simple blood test. Now we are all, what are we focusing on? Neuroprotective strategies to reduce neurodegeneration. How a simple blood test? So now if you think about that, early identification of patients with hyperglycemia or hypercholesteremia, what can we do? A simple drug can reduce the risk for neurodegeneration, can reduce the risk for neurodegeneration. So that is the big picture that I want you to understand. Okay. So now we are having some strategies to reduce the hyperglycemic signal. So again, you can think about that. What is happening in hyperglycemia? Okay, you have a possibility of two: decrease insulin and or resistance to insulin. So that can cause all these neuropathology. So that's where we predicted predictable hematological markers associated with cognitive decline. Okay, we use two markers of cognitive impairment. One is hyperglycemic marker, and number two, I don't know whether you have heard about this word, chemo brain. It's a very popular scientific word. People who are having cancer in India, we have numerous patients with cancer, and when they are administered with chemotherapeutics, what are chemotherapeutics? Drugs are chemicals, pharmacological approach to reduce this growth of the cancer cells. These chemotherapeutics, yes, they can kill the cancer cells, but all these drugs, most of these drugs, what do? They cause cognitive impairment. So that is the reason we want you to understand why I'm coming with the neuroprotective strategies. Okay, good enough. And now the last one before I wind up is genome. So as I told you, now most of you scientists, I know you guys are very powerful and you know a lot of people. You are having now, because of COVID, most of the hospitals have machineries or scientific equipments to do some lot of typing, genomic typing. So now we can use it for our research. If you can identify a particular gene in a pool of people, okay, we can do a lot of pharmacoepidemiological study, pharmacogenomic study, pharmacogenomic epidemiological study. So you can see that in 1990s, there was only one gene isolated with Alzheimer's or associated with Alzheimer's disease. But however, now, I think we have around 50, 70 genes associated with the Alzheimer's disease. So now we can find out certain resistant genes or certain gene that increases the risk for Alzheimer's disease. And with your help, we can do that in the population in a particular region of India. So think about that, because we have the tools now. So the next one is to identify the genes associated with neurodegeneration and have the patient alerted at a very young age. Okay, you have these particular genes, so you're at a higher risk. So just we want to make sure that patients, so that they can have non-pharmacological approach, a proper exercise, proper diet, which can, what can we do? Which can reduce the neuronal loss. So that's the reason, again, I'm coming with the genomic approach. And then currently we call this a single drug approach is gone. Most of the diseases for neurodegeneration, we are going to go with multi-targeted ligand, which means a drug that has multiple pharmacodynamic effects. Okay, that's the whole idea. A drug does not have one pharmacological effect, has a multiple pharmacological effect. I'm just going to show you that. Okay. So whenever you think about currently for Alzheimer's disease, we only have two drugs, a cholinesterase inhibitor and NMDA antagonist. So now what is the pharmacy coming up with? They are coming with combination of two drugs. When you think about Alzheimer's disease, when you think about Parkinson's disease, what are they trying to do? Exactly. They're not using levodopa anymore. We use levodopa and carbidopa. But recently, what do we have? We don't only have our aromatic amino acid inhibitor, we also have a COMT inhibitor. So currently a levodopa formulation comes with levodopa, entacapone, and carbidopa. So think about the picture. So we are going for a multi-targeted drug therapy. So why am I bringing this concept? When you think about Parkinson's and Alzheimer's disease, there are numerous things that's different, but the neurodegeneration occurs with oxidative stress, apoptosis, mitochondrial dysfunction, inflammation, excitotoxicity. So what can you do? So now let's be very clear that we make sure that we have prophylactic drugs, which has antioxidant effect, anti-apoptotic effects, a drug that can enhance ATP like coenzyme Q10, anti-inflammatory drug. Aspirin a day keeps the heart away. You all know that, right? An aspirin, low dose aspirin really is good. It can decrease the death or morbidity and mortality associated with the cardiovascular problems. So then excitotoxicity, think about an NMDA antagonist. Excess glucose acts on the NMDA, causes excitotoxicity. So you want to block that. So let's think about these kind of strategies. Again, as I told you, nowadays, with most of the drug therapy, it has become a multi-target directed ligand. So you want a ligand to have anti-inflammatory drug, decrease beta secretase, has antioxidant, okay? So that it can be used in the treatment of Alzheimer's, which is a neurodegenerative. So in a big picture, I'm just giving you one or two strategies how we can reduce neurodegeneration. I want to thank all my colleagues and my students. Especially, I want to thank Dr. Jaybalan sir for inviting me to give this particular lecture, and also the dean. Thank you. I'm ready for questions.
Thank you so much, Dr. Murli Krishnan. And it is a very wonderful because you have in-depth knowledge of the neurodegenerative diseases, and especially the way you have explained to the students the possible approaches to the neurodegenerative diseases is very good, and it is simple and understandable. As a pharmacologist, I also got to know few things out of your approaches, and I think your approaches are and will come a new direction in the treatment of neurodegenerative diseases. And we would like to have some future collaboration with your university, and because in India we have few things, but we are hampered with the new technology, new softwares, in silico softwares and other studies. But definitely the way you have explained about the natural ingredients which are being used for the treatment or have been targeted for the neurodegenerative diseases, especially the Parkinson and Alzheimer disease. So that is a and how we can approach the bioavailability, we can enhance the bioavailability, and the way you have explained what is oxidative stress and how this oxidative stress leads to various things which can be a problem creator for this neurodegenerative disease. So I thank you so much for your very extensive and very elaborative lecture, which has given our students insight about the approaches to the for the research in neurodegenerative disorders. Thank you so much, sir.
Thank you, sir. Anytime, sir, please call me. I'll be very happy to have collaboration, help you out anytime.
Thank you very much, Dr. Arun Garg, sir, for your sweet and short comprising of this session. And I also would love to hear from Dr. Kamal Singh Rathod, sir, who is in the audience side. Sir, you are right from the beginning, Dr. William Carey and Dr. Kamal Singh Rathod, two eminent professors of pharmacy side. Sir, can you please unmute yourself and share your views on this lecture?
Sir, this one is very interesting, and to know the details in very simple manner, sir. And we are very happy to learn something from this seminar, sir, lecture. Thank you very much for arranging this this type of program, sir.
Thank you, Dr. Terry, sir.
Sir, good evening.
Yeah.
And thank you so much for mentioning about me. I was enjoying this lecture. It was really very informative, and I hope all students really got something from this lecture. thank you so much for conducting such a nice, wonderful lecture, sir. It's wonderful, nice.
Thank you. Thanks for your opinion, sir. And there are so many senior people watching this session. Actually, we are doing this after a long time because our students are doing their examinations now, and Dr. Arun Garg sir was motivating me to conduct the events continuously. So because of this motivation, we started again this conventions with the collaboration of NIMS Institute of Pharmacy under the guidance of Dr. Arun Garg sir. Thank you, Dr. Arun Garg sir, for this support and motivation. And I love to thank always Dr. Muralikrishnan, because in one way, in one word, he readily accepted my request to speak among us in this evening for us and early morning for him. He's sitting around in early morning hour, 6:30 a.m. So thank you, Dr. Muralikrishnan, for sharing your research and models for understanding, or for making us to understand this concepts of neurodegenerative diseases and prevention of this kind of neural deaths.
Devalanji,
Sir.
I would like to say that the students would like to have more about this in silico, what are the various software they are using, and then how they are coming up with this pharmacokinetic and pharmacodynamic finding. So if they can once again, or in vivo techniques, what they are using it, especially the in silico techniques where they use the computers and all those softwares, and then go for the ligand binding studies and other things. So if that can be explained in another extension lecture, we would like to hear once again with Dr. Muralikrishnan.
Yes, sir. Thank you. I'll definitely. He's also hearing the same request. So he'll be very ready to. I was actually asking him to come to India for a conference in Alwar, but due to this pandemic situation, we were prevented to meet for the past 11-12 months. So we are eagerly waiting for the opportunity. So as soon as, otherwise we will do one more session with Dr. Muralikrishnan and Dhanasekaran one more time with the students. This is actually to inform to our students and to bridge where the researches are being conducted in other countries, and where are we in the neuropharmacology. We are studying only about the introduction of this neurodegenerative disorders. That's all. For GPAT and all, some of the students will study some about some markers and all, but they are not going in deep about the researches on this neural problems or neurodegenerative disorders, as it is not a very common disease in India nowadays. So we are not taking much care on that. So we wanted to make our students to understand and bridge the gap what we have to learn about this disease. And I think we are approaching towards our aim. So we will do it in another 1 or 2 sessions. A complete picture can be given to the students so that they can also easily understand and do this kind of researches in our laboratories. So with this, I'd like to proceed to the next part of this program. That is our 1 minute. So now this is the vote of thanks. The vote of thanks will be given by Dr. Rajesh Sharma, Professor of Pharmaceutics in Alwar Pharmacy College.
Good evening, everyone. Dr. Rajesh, Associate Professor, Alwar Pharmacy College. It's my privilege to propose vote of thanks in front of you. First of all, I would like to thank Dr. Murli Krishnan Dhansekharan sir, Professor Pharmacology, Urban University, USA, for such a wonderful presentation on novel strategy to prevent and treat neurodegenerative disease and pharmacological approaches. Thank you, sir, for sparing your precious time with us, and I hope the student and attendees definitely benefited from this presentation. And I also thank Professor Dr. Arun Garg sir, Dean, NIMS Institute of Pharmacy, NIMS University, Jaipur. And I also thank Dr. Kamal sir and Dr. M. William Carey sir, Dr. Narendra Sinsinwar sir, and my sincere thank to Professor Dr. G. Jaybalan, former Dean, Faculty of Pharmacy, Rajasthan University of Health Science, and Principal, Alwar Pharmacy College, for giving me this opportunity. Thank you, sir. Thank you, and also thank you all the attendees for this webinar. Thank you very much, sir. Thank you.
Thank you, sir. Sir, Dr. Murali Krishnan, you want to say, you want to convey any message to us?
Once again, I sincerely want to thank you, sir, for inviting you and Dr. Garg for inviting. Thanks for the great opportunity, number one. Number two, I'll be very happy to do more seminars with you in a very nice way. And when I come next time, once the COVID is over, definitely I'll be there in Jaipur and all that. Thanks for the kind invitations.
We are waiting for it.
Thank you. Thank you.
Welcome. Thank you very much, one and all. Thank you, all eminent personalities and my dear students. Thanks for attending this session. And this is also live streamed in the YouTube channel of Allur Pharmacy College. So you can have a look at any time later. Thank you. Thanks all of you. With this, I would like to end up the session with the permission of the chair. Sir, can we proceed?
Yeah, you announced that we are going to have more sessions on industry institute interaction, clinical research, and some other topics in a series of this
in the next week
collaborative programs.
Yeah, in the next week we'll have more and more programs with the guidance of Dr. Garg. There are so many resource persons are identified, and I think you'll be getting benefit out of it, definitely. Thank you for this session. Thank you, Dr. Murali Krishnan, especially, and your family for allowing you to wake up early in the morning and making noise in the bedroom.